This one? This will be on the other side.
So, if you have to come in on thing? Yeah. So rather than doing the quiz in class today, we are going to do it as a Moodle quiz, just so we don't take time. And I emailed you about this already, but just as a reminder, the quiz is going to open just after class today, and it is due on Sunday by 11. And it's gonna be essentially set up like a pre-lecture assignment, except that it's gonna be you're gonna have a time limit, so it's 30 minutes. You will have 2 attempts because sometimes Moodle mistakes. And so just in case something goes weird with an attempt, you'll have another attempt. But you're not gonna know after the first attempt how many, what's correct and what's incorrect. Okay? So this is essentially set up as a real, quiz. So you'll have 30 minutes to do it. So I would suggest you study beforehand because you won't have a lot of time to look stuff up. So let's see. That means you have to do the 2 attempts within 30 minutes. Okay? But you can do that anytime between now and Sunday evening. And also don't forget that you still have our pre-lecture Moodle assignment due on Sunday as well. And the other thing I just wanted to mention was discussion next week will be a little bit weird just because our discussions are Wednesday and Thursdays, and the exam is Wednesday night. So I don't do exam material in discussion this week because it wouldn't be fair for Wednesday discussions to get that review, and then Thursday discussions, it'll be all over, right? So happy to have discussion on Wednesday and have discussion on Thursday. So discussion material for next week is going to be the beginning of the material that we're going to start after the signal transduction section, which we'll probably get to on Wednesday. So I apologize because I know that the day of the exam, if you have discussion, you're going to be wanting to think about exam material, but for discussion where the material is going to be past exam material. Does that make sense? So we'll be moving on to discussion. We also do have quiz. We also do have class on Wednesday. Okay? And again, that material will not be on the exam on Wednesday. Okay? For the next exam, actually, for exam 2, it's on Thursday night, so we won't have this problem. So for that week, that discussion material can cover exam material. But for Wednesday night exams, it's not really there. Okay? Alright, so this is all of the exam 1 information. So there's a review session on Monday night with the TAs, 5:30 to 6:45, and that's for 1 and 4. There's a review Tuesday night with me at same time, same place. Wednesday night is the exam at 7 and now for 20. If you have a class, exam, work, family, religious, personal conflict for Wednesday at 7 o'clock, there are alternate exam times that day at 10 a.m., 2:30, and 4. You do have to have a valid excuse for needing the alternate time, and you do need to sign up. And I have a sign-up sheet up here, so you can do that for class. Okay, and so here are the times in the rooms. All right, any questions for now? So, we were working on beginnings of concepts in signal transduction. Okay? And so on Monday we finished with this question about whether the signal was present or not. Okay? And remember that each of these components, that the signal has to have been received initially, but once the signal cascade, the transduction cascade is heard, each of these components is regulated separately. Okay? So once signal arrives, you get RTK phosphorylation, TK1 phosphorylation activation, TK2 phosphorylation activation, TK phosphorylation activation, and then activation of transcription. Activation of transcription is considered the outcome of this pathway. Okay? And so the question for this one was, is signal still present? Okay? And really, once you get someone comes along and activates the pathway, and you get transduction of the pathway, signal in theory can leave. Okay? And as long as the components of the pathway are still active, then you're still going to get outcome of your pathway. Okay? So that was sort of the main point of that question. Alright, so why don't you try this one, this clicker question. This is a multiple choice. So basically, you analyze your cells and it's so analysis shows that the cell has a mutation in the gene for TK2, such that the protein does not exist. Does this pathway still function?
That's not good.
Oh, that's right.
And this one is. Do you know what to chat about?
The regular Wednesday 7 p.m. I think they were. Right?
They're on Moodle too.
Yeah, and all. Alright, you guys done? You in? Yeah. Okay. And we'll turn it off then. Okay. So what do you think? What do you want to tell us what you answered and why?
Well, isn't the protein required for that phosphate to continue the chain? So if the protein isn't present to receive the phosphate, then wouldn't it just stop there?
So TK2, you mean?
Yeah, if the kinase isn't there, then wouldn't that mean that the whole process should just stop because the phosphate can't transfer onto the TFA? Even though the next molecule necessarily isn't a protein.
It is a protein.
Or an enzyme.
It's not an enzyme.
There we go.
Yeah.
So it's
Transcription
protein, but not an
factor,
enzyme.
right, so transcription factor is a protein. And its job is to bind to DNA. So it's definitely a protein and it has a job. It's just not a kinase, it's not an enzyme. Yep.
I could tell maybe because if TK2 is the only phosphatase that can activate TFA,
Kinase.
the kinase
Yeah.
that can activate TFA,
Mhm.
would be a true statement.
Mhm.
However, if TK1 can also activate TFA without like a middleman,
Okay.
so to speak,
Mhm.
then it would work. So I was going to ask you that question, like, are we assuming that this is the only?
Right. So that's the question.
Yeah. Are we? No, I'm saying are we assuming?
That is the question.
Oh.
So that is sort of the main concept to this question. Right? So can TK1 phosphorylate TFA? And does it make sense to have TK1 be able to phosphorylate TFA?
No, it doesn't because then you wouldn't need a TK2, right?
Right. And so, I mean, really, I showed the Rube Goldberg, like, in whatever thing video, because the whole idea behind that is that you need one thing to happen before the next thing to happen, before the next thing to happen, before the next thing to happen. You can't just skip steps, right? And the cell regulates, this very, very carefully, right? So this gets back to the concept of, these things work in very specific ways, and kinases have very specific substrates, okay? And so so TFA is not a substrate of TK1. You know, and so TK1 has a substrate, its substrate is TK2. And TK2 has a substrate, and its substrate is TFA. Right? Anybody else? Questions, comments, arguments?
Why does the cell have multiple kinds? You have to delivery service.
So, right, and so part of the idea is why have multiple steps, right? Why not? There is one pathway called the JAK-STAT pathway, where the transcription factor actually binds directly to the receptor and gets activated, and then just goes to the nucleus. Okay, so why have multiple steps in your signal transduction pathways? Yeah.
If it's too simplified, then like a mutation can be like all of it, or unfortunately, one step is mutated.
Okay, so having multiple steps potentially might allow for mistakes, to not cause as much trouble.
Yeah.
As you were saying, yeah. So, do you remember going back to the original diagram I showed you guys of all the signal transduction, not all the signal transduction pathways, but many signal transduction pathways, right? So what's one of the things that those signal transduction pathways do with each other? They talk to each other, right? So if you have multiple steps in a signal transduction pathway, then you can get some of those steps, some of those kinases, to talk to other kinases and other proteins and other pathways, right? The other idea that I don't really talk about too much is the idea of signal amplification. I really hard time saying that word. Signal amplification, okay? So you receive one signal on one receptor, okay? But maybe that receptor can talk to a bunch of TK1s, and maybe those TK1s can talk to a bunch of TK2s, right? So you have, even if you only have one signal molecule, and this is actually really important, understanding that there's not, so this is where we start building complexity into our model, okay? So the idea is that rather than it looking something like this, that it looks something like right, where you have one signal coming in, but you have multiple downstream components getting activated, okay? And this makes it, and why do we need this? Because cells are kind of big, complex places, and proteins move around the cell pretty randomly. Most proteins do, some don't. Some actually go along like trivials. But a lot of proteins move in the cell really randomly. So how is TK2 in this big space supposed to find a TFA? Bump into it, find it, and phosphorylate it. Right? So it turns out that there's lots of TK1s, and there's lots of TK2s, and there's lots of TFAs. Okay? There are populations of these proteins. Yep?
So basically has like a hierarchy?
Not necessarily a hierarchy. It's the concept that there isn't just one of each type of protein in the cell. That there are pools of proteins. And one of the things that the cells regulate is how many of those proteins there are at any one time. You know, from 0 to thousands. Right? And so signal amplification makes it easier such that one signal being received by one receptor can actually make it to the nucleus, especially if you have multiple steps that need to occur. Because that means, again, that TK1 has to find TK2 in the cell. And relative to the size of a protein, the cell is a pretty big place. Okay? And so if you have, say, 1,000 TK1s and 10,000 TK2s, and 20,000 TFAs, then, and because you're working with kinases, and kinases are enzymes, which can do their enzymatic activity over and over and over again, right? So, so we started with one of each protein, and now we're moving into building a little more complexity and to start understanding that the there is more than one of each protein. Okay, and this is important because we're going to start talking about how you modulate these pathways. And one of the ways you modulate these pathways is by modulating the amount of each protein that's there. So if you want to say, if you've gotten signal transduction, but you want to down-regulate the signal transduction pathway, what might you do? Make less or degrade the ones you have. So get rid of the ones you have. Right? So if we have 1,000 TK1 molecules, and one of the ways that you can downregulate signal transduction is by degrading TK1. So cells have ways to get rid of their proteins. So we'll talk about that today. The other piece of complexity that I want to build into this is the idea, and just, when I ask you a question about this pathway, I'm asking you a question about this pathway, okay? But keep in mind that you were talking about that if you make a mutation, that it might be less likely to have a really devastating impact. Okay? So one of the other ways that cells do this is to make redundant pathways. Okay? So it could be that you have two pathways that result in the same outcome. It's not working very well. Okay? Now, in terms of our pathway, we haven't really, detailed that. Okay? But just understand that this does exist in cells where, especially for processes that are really crucial, sometimes there's more than one way to get to it. Okay? So one example is, we'll actually see a slide in this, a review about this, but exit from mitosis. Right? So cells go through the cell cycle. They go through DNA replication, they go through S phase, you guys learned this in high school or previous biology class, right? And then eventually they go through chromosome segregation and phase cytokinesis, right? So, and these are all signaling pathways not signaling pathways, but pathways within the cell that tell cells how to go through the cell cycle. And there's one called exit from mitosis. So it tells the cells, okay, you're segregating your chromosomes, it's going to exit mitosis. And so it actually turns out that there are two pathways called MEN and SYN. MEN stands for mitotic exit network, and I don't know what SYN stands for. There's two redundant pathways, and you can basically knock out one pathway and still get exit from mitosis. And so there was a professor that I knew who studied this and actually did a lot of work showing this, and she wrote a review in 2001 called Men and Sin, What's the Difference? Right? Anyway, so redundant pathways. Okay? And so in theory, and this is really, cells create, well, I don't know, really, cells don't really create redundant pathways, but redundant pathways exist, as far as I can tell, for really, really, really important functions. So that if you do screw up one of them, that you have another one. Right? So if you make a mutation in one pathway such that it doesn't work anymore, that you have another pathway. Right? So in theory, if this did have a redundant pathway, might you still see activation of TFA? Sure. If there was another kinase that could phosphorylate it and activate it. Okay? And that's a big if. Not all pathways have redundancy. So. Okay? Good. I like how you guys are thinking about this. Okay. But yes.
In all pathways, is there only TK1 and TK2?
No, so we're using really generic names, and it's really, there are many, many different kinds of signal transduction pathways, and the ones that's modeled after has actually 3 kinases. So if you guys end up looking at which will, so one of the slides and a couple of slides is actually looking at the real pathway. And if you look up the RTK MAPK pathway, so RTK stands for receptor tyrosine kinase, which is the receptor. right? MAPK stands for mitogen activating protein. So mitogen is another word for growth factor. So these are the growth factor pathways. These are the pathways that tell cells to undergo cell division. And if you look up MAPK pathway, they actually kind of have a lot of names. So the other, sometimes the name that they also sometimes use is MAPK pathway. This is why I use generic names, because scientists like to give lots of names to the same thing. But the generic names for the three kinases that actually function in this pathway are MAPKKK, MAPKK, and MAPK. Okay, so basically activation of RTK eventually leads to the activation of MAPKKK, which leads to the activation of MAPKK, which leads to the activation of MAPK, which then leads to the activation of the transcription factors in the pathway. Okay, so this is like the real one. Okay, and one of these guys is called MEC, and one of them is called ERK, and I can't remember what it is. All right, do you know what KKK stands for? Other nations of the plan? So what were they thinking when they made these names? I mean, really?
Apparently it starts from 4 K. Apparently it starts from 4 K. MAPKKKK.
Oh. Yeah, most of the ones I know of have. I mean, there are different variations on these pathways in different cells. The ones I know about have 3. And the K actually stands for kinase. So it's like MAP kinase, 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 phosphorylates MAP kinase, kinase, which phosphorylates MAP kinase, which then phosphorylates transcription factors. Okay, it's really, really annoying. But I mean, that's, you know, and these are actually, again, generic names. So a lot of different organisms have variations on this pathway, and the gene names are different in different organisms. So that's why we just do sort of this simplified generic pathway to get at the concepts. And then if you ever advance onto a class where you're actually learning like signal transduction pathways, then it won't all seem really foreign. Like, what kinases? What are you talking about? Did what? So there's lots of kinases involved in signal transduction pathways. Go back. Okay. So like this is PI3 kinase. I can't remember AKT. PKA is protein kinase A. Here are your MAP. So this is MAP KKK. This is MAP KK, and these are 3 different kinds of MAP Ks. Okay, in your RTK pathway. This is a kinase. This is a kinase. So yeah, there's lots of different kinds of kinases. Yeah.
So I know that receptors are signal specific.
Yeah.
But are signals pathway specific? Like could a signal, because they're all interrelated, so could one signal have effect like on one pathway and then that pathway has another pathway, it would be because of that signal, or is it just a picture?
So signals are receptor specific. Okay, and so this gets into molecule protein interactions are very specific, just like kinase substrate interactions are really specific. Signal receptor interactions are really specific. Okay, and so what you're asking really depends on who the receptor interacts with and who the downstream signaling components interact with. Right? Can a signal affect more than one pathway? Okay, signals are specific to receptors, but then once you get down beyond to receptors and then to the signaling components below the receptors, they can spread out and talk to each other. Right? Okay. Okay. So now that we've learned a few things about signal transduction, we're going to talk about how you modulate. We've learned how you turn on output or get output or outcome of the pathway. But how might you modulate it, and why do cells want to modulate signal transduction pathways? Do they want them to be on all the time? Right? They have to respond to their environment, and their environment is constantly changing, right? And so their signal transduction pathways have to be constantly changing, whether they're turning them up, turning them down, turning them off, whatever. Okay. And so we're just going to talk about a few ways in which cells might modulate their signal transduction. Okay. pathway that we've been talking about, okay? And say it's in a particular stem cell type, and let's say that upon transduction of the signal, so signal transduction and output, which is changes in gene expression, let's pretend now, since we're layering in complexity now, that one of the genes turned on and made into protein is another kinase. Okay? And we're going to call this kinase TK3. All right? And we're going to say that TK3, which is a gene activated by TFA, goes and phosphorylates TK1. Okay? So the question is how would that change the behavior of the pathway? Make this a multiple choice. You have to just pick one. Where's that gone? It's not behaving. Yeah, that one.
That's the way I was doing it. Just because the allergy to do these things.
I just pulled up the page so I can keep a list of relative links to put in the description.
Yeah. So, EGF is
If you go up and down, it's probably not going to be the same. If you go down, it's like, is it that thing? Is it like, how cold is too cold? One of like,
One of these things is that we should remove the variants exclusive.
I think they're more interesting.
YouTube exclusive extra commentary.
They make some particles too. No, text box.
yeah.
I think Dave only has 3 right now, so people.
Yeah. Okay. Great. Are we done? You guys done? All right. Okay. There we go. All right. Tell me what you think. What you picked. I don't know what E stands for, but yeah.
I picked B because it creates a positive feedback loop.
Okay.
Such that you don't need this signal when TK3 already activates TK1, so this keeps going.
Okay, so you're saying that basically signal would be received, transduction would occur, gene expression would be turned on, TK3 would be made, it would go and phosphorylate TK1 and serve as an activator of TK1, which would then just continue the pathway down again and back up and down again and back up and down again and back up. Alright? You're calling that a positive feedback loop. Okay?
you said before that you don't need single vector, so protein
Yeah, okay, so you're saying it wouldn't change because you think the signal left, it'll still keep going anyways. Okay, but this sort of guarantees it, right? What if there's a phosphatase that comes along and dephosphorylates TK2, right? Then this would sort of guarantee the continued activation, right? As long as you're getting output from the signal transduction pathway, you're getting activation of the signal transduction pathway, right? Positive feedback. Anything else? Anybody that disagrees with that? Does phosphorylation always have to be an activation event? No? So, on the assumption that it is an activation event, that's fine. Now, what if TK3 phosphorylation of TK1 were an inactivation event? Okay. So, if TK3 inactivates TK1 by phosphorylating it, so it can phosphorylate it, say, at a different site on the protein. Okay. So now, with this signal present, what's going on to TK1 then? I mean, what is TK1 experiencing? What? No, because we're saying TK3 is an inactivation event.
Oh,
TK3
yeah,
phosphorylation. So who's activating TK1? Signal, right, through our TK. But TK3 is inactivating TK1. So what is TK1 going to do?
It's going to stop the signal.
Once the signal goes away?
If the signal's still there, wouldn't it just like counteract TK3?
Right, so if you got TK1, alright, and you've got, on the one side, you've got signal working through our TK, which is an activating event, right? I'll put little pluses. Alright, and on the other hand, you've got TK3 phosphorylating TK1, and I'll make minuses because that's an inactivating event. Okay? Now again, keep in mind TK1 is probably not a single protein all by itself, so there's pools of TK1. Alright? And so these pools of TK1, on the one hand, they're getting activated by our TK, and on the other hand, they're getting inactivated by TK3. Okay? So at this point, would you expect to see decreased signal transduction? Okay? So as compared to when there's no TK3 around, which is like when the initial signal comes in, okay, TK1 is just getting activated, all right? But once you get signal transduction, and then you get production of TK3, if this were an activating event, you would expect to see decreased activation of TK1, decreased activity, shall we say. And it's always going to be this, you know, at this point, unfortunately, you have to get into chemical rates and kinetics and all that stuff, which we're not going to get into. But just understand the concept that this would modulate the pathway, right? It would modulate the pathway. Now, what happens if signal goes away and that RTK becomes inactivated by something? Okay? So now what's going to happen?
TK3 is going to, well, yeah, TK1 is going to stop.
Right. So TK1 will become inactivated, eventually TK2 will become inactivated, eventually TFA will become inactivated, and eventually you'll stop the pathway. Okay? Alright. I've got one more thing to throw at you, which you're just going to love. Okay? Alright. We'll talk about feedback loops in a second. We'll talk about circuits. initially, and activates TK1 and activates the pathway, how much output do you expect to get? So here's signal. Alright? And at some point, you'll get activation of the pathway, so the amount of output you get will increase. Right? Okay. But then, as a result of the pathway, you get some sort of output, and this could be, we could call this a kinase, or a phosphatase, or really anything that has a negative effect on TK1 or some other component of the pathway. Okay? So, negative effector, we'll call it. Okay? Now let's pretend that that negative effector wins. Okay? That it just shuts down TK1. Okay? So then how much output are we going to get to over time? If we shut down TK1. So eventually we'll go back down to 0, right? Or low anyway. Okay? Now, but as a result of shutting down the pathway, what are we going to stop making? The negative effector, right? So now we're not making him anymore, and let's assume that he doesn't hang around. Okay? And then if the signal is still there, what's going to happen?
It's going to continue.
Right?
It will be oscillating.
We'll go back up. But now you're getting output again. And so what are you getting output of? Crap, you're getting output of your negative effector again. Okay? And now that's winning out, and it's gone. Okay? So what does that look like? A firefly? On oscillating output, right? So, also known as an oscillating circuit. Okay? These things happen. These are actually oscillating circuits control your circadian rhythm. So it controls your sleep-wake cycle in your body, in your brain. Okay? Oscillating outputs control, does it control firefly blinking on and off? Okay? So these things exist, and they are important. They're what make us go to sleep at a certain time and wake up at a certain time. Okay? So that's an oscillating circuit. Okay. So you guys should have a sense of how that works. Okay? All these things that we've been talking about here are examples of feedback loops. Okay? The definition of a feedback loop is when you have a linear pathway, meaning, one thing affects the next thing, which affects the next thing. Okay? So you have a linear pathway, and the definition of a feedback loop is that a downstream component of the pathway affects an upstream component, regulates an upstream component of the pathway. Okay? And again, really, really, really common mechanism that cells use to control themselves. Okay? Like, so for example, when cells are making an amino acid, right? Building block of proteins, sometimes used as an energy source, a nitrogen source, whatever. Okay? There's a biochemical pathway to make the amino acid. How do cells know when to stop making that amino acid? What is the signal? When there's enough amino acid. So if the amino acid itself controls its own production, then when amino acid levels get to a certain point, then it downregulates the biochemical pathway that makes it stuff. That make sense? Okay? So, again, really common mechanism that cells use to regulate metabolic functions, regulate signal transduction pathways, really anything where you have a linear pathway of events. Okay? And so it needs to be able to turn it on, right? But then it also needs to be able to turn it off. It needs to know, or just to downregulate it, it needs to know when it should do that. And so if you're making something, right, like, I don't know, like a nucleotide, which is really important in the cell, right, or an amino acid, you have biochemical pathways that make that amino acid, okay, And if you have the amino acid itself go back and regulate one of the enzymes in the pathway, right? So when the levels of this amino acid get high enough, it goes and it shuts down one of the enzymes in the pathway that makes itself. It's beautiful, I tell you. Okay? Design-wise, it's cool. Alright, and so what I'll say is negative feedback loops are a lot more common than positive feedback loops. But positive feedback loops certainly do exist. And actually, I don't know exactly how this works physiologically, but when you go into labor, that is a positive feedback loop. Somehow going into labor makes you go into more labor.
How would a positive feedback loop stop? Would it just exponentially increase?
So how does, well, it would have to stop. That's a good question. How would it stop? You would have to stop it somehow. Okay, so you would basically have to get rid of one of the components of the pathway, right? You could get rid of the guy that's coming back up.
So it would have to be removed from like
You
an
have to
external
remove
source or
something
something.
out of there, yeah. Or else we'll just keep going, right? But we'll talk about this, okay? So don't worry too much about the mechanism here, and worry more about the big picture idea, okay? How do you remove proteins out of the cell once they're made? You send them to the garbage disposal, also known as the proteasome. And honestly, the proteasome kind of does look act like a garbage disposal. It has this like a barrel-shaped structure, and you feed the protein into the barrel, and it basically chops it up. Okay, chops it up into little pieces. All right. And do you think this is a regulated process in the cell? Would the cell want it to be a regulated process? Yeah. You don't want to just like go around like cutting up more proteins, right? Some of them you actually need, right? So basically all these guys up here that we won't really talk about are enzymes that regulate this process. Okay? And basically what these guys do is they tag the protein that's supposed to be cut off. Okay, they tag it with a molecule called ubiquitin. It's a little protein, tiny little protein tag, kind of like a bead. Okay, so the proteasome, the garbage disposal, needs to know who it's supposed to be chewing up. Okay, and these little ubiquitin molecules tag it, tag the substrate for destruction, basically. And these E1, E2, E3, they're called ubiquitin ligases. It's not that important. They're the ones that select the proteins for destruction. Okay? So that way, if you are in the middle of a signal transduction pathway and you need to shut something down or modulate it in some way, the cell can activate ubiquitin-mediated proteolysis of one of the proteins that needs to be gone. Okay? If the proteins in the pathway are activated by phosphorylation, who else could you activate to downregulate the pathway? Who takes phosphate off? Phosphate, right? So cells also have the option of not getting rid of the protein, but of dephosphorylating the protein and keeping the protein around. It costs the cells a lot of energy to make protein, okay? Which is why they modulate protein after they've made it, okay? So degrading protein is a decision that the cells don't make lightly, because if they have to remake it, it takes a lot of energy to remake it. Okay? Alright, and really quickly, the other thing that I want to talk about is one of the other ways to make sure that a cell does not receive a signal any longer is to do what? Eat it? Eat the signal? Eat the signal. You can get rid of the receptor, right? Alright, so there's another process to keep in mind called receptor-mediated endocytosis. Alright, and if you remember, endocytosis is a process where, do you remember the video that I showed you where the neutrophil was running around after the bacteria and eventually ate it? That was endocytosis of the bacteria. Okay? So basically, a vesicle is formed and essentially pulls the receptor back into the inside of the cell. Okay? And so if you want to modulate the ability of the signal transduction pathway, you can just get rid of the receptor off the cell surface. Right? In receptor-mediated endocytosis, there's basically two things that cells can do. They can either degrade the receptor if it decides that it doesn't want it anymore, or it can rip the signal off of the receptor and recycle the receptor back to the cell surface to receive more. Okay? So again, this happens, it's important, it modulates signal transduction, it modulates the pathway. Alright? And on Monday we will start with this question that I think it's called